I'd like to introduce today's CCMB speaker, Stewart Silfron from Icahn School of Medicine at Mount Sinai. I've had the pleasure of getting to know Stewart over the last couple of years as part of the Motor-Pack project, molecular transducers of physical activity that we both are PIs on. One of I guess 20 grants are funded as part of this larger NIH Common Fund project. Stewart did his undergrad in comparative literature at Princeton University. One of the things I got to know about Stewart is that he's a well-read guy, and we've discussed books on several occasions, and now I know why. Then he went to medical school at Columbia University and did an internship in internal medicine and residency in neurology at the Mass General. And then he did a postdoctoral fellowship at Mount Sinai, and then stayed on and actually rose through the ranks where he's now professor of neurology and neurobiology at the Icahn School of Medicine. He's held numerous administrative positions at Mount Sinai, including serving as chair from 2009 to 2015, and now is the director of the Center for Advanced Research and Diagnostic Assays at the medical school. His basic and translational research has been mostly focused on pituitary and related activities. He has a strong translational bent to his research career, and it has led to over 200 publications in some of the highest quality journals around. He's also had a number of patents and contributed to the development of a couple of drugs in his career, which is exciting for any clinical translational researcher. Stewart is an assay genius, and he's going to talk to us today about his work with the U.S. Navy and looking at the spread of the COVID virus. He's been telling me a little bit about this work over the last couple of months, and that's one of the reasons I twisted his arms to come and give us a talk. So Stewart, I want to give you the time you deserve. Thanks for coming, as it were, to University of Michigan. Looking forward to your talk.
Thanks a lot for that very nice introduction, Chuck. Let me find the slides here and make them full screen. There we go. So I guess this graph on the right is the increase in COVID-19 diagnosed cases and the increase in COVID-19 papers. And there's almost exactly 1 to 400 cases, paper to cases worldwide. So and it's tracked quite consistently through the pandemic with no lag, interestingly enough. So, so many of us have become COVID-19 researchers, and in my case, I've also ended up getting deeply involved in clinical studies for the first time in my career. And so I'm going to take you a little bit through how we got there and what we've learned. And it evolved out of this DARPA program involved in, which works on blood signature development for infection. And then we were asked to support and help stand up this Navy program of new Marine recruits. I'll take you through. And then the reason we got involved in this, and DARPA got involved in this in large part, was to try to generate samples for a diagnostic test for COVID that might be earlier than the PCR test. Although I think it was really designed for a different country that did not have so many cases, that our test positivity is running at 10% much of the time. So I think a more sensitive test is not necessarily needed. The idea of this was for trying to facilitate limiting quarantine and ruling people out from being infected. In any event, the DARPA program is called ECHO. It was started in May 2019, and the idea is to try to use epigenetic characterization to predict someone's exposure to specific infections, when they were exposed, and also to chemicals and radiation and other components of weapons of mass destruction, but significant emphasis on infection, and also to develop a portable device that would work in the field on a small blood sample, which I won't go into that aspect of it. And in part, Chuck and I are involved in MotorPack, and MotorPack has been so slow and so lumbering in getting things going, getting samples generated. It's an incredible study, but it's very complicated. And so when when I saw the announcement for this program, I thought, gee, this is something that maybe we could do faster, as sort of a mini MotorPack like study that we'd have control over more of the aspects and not have to deal with as many committees. But it was ended up being a little faster than I would have expected. So the approach that we follow is it's a consortium we've assembled of investigators at various institutions, and we take BBMC samples, cryopreserved, and put them through data generation, also integrated with any public data we can obtain. We've developed some new ML tools for building models, and we identify a diagnostic signature, downsample it, and then try to validate it on independent data. And to give you kind of a couple examples of things we got to before COVID hit, this is a small data set of single cell RNA-seq and single cell attack-seq on the same samples of individuals who had been infected with MRSA or MSSA. And Xi Chen, and I know Olga Troyanskaya spoke here a while ago. I work very closely with her, a number of people we're collaborating with in her group. And Xi Chen took the differential expressed enhancers and mapped them to differential expressed genes via GeoPad and their enhancer prediction compendium, and then use that to train a classifier. It's not a huge data set, but you can get nice segregation of the MSSA and MRSA samples from their chromatin accessibility, differential accessibility, and differential enhancers winnowed down to this group. We also did a study that's still ongoing of organophosphate exposure, pesticide exposure in migrant field workers. That's being led by Lisa Satterwhite at Duke. And in this case, we've only studied the RNA-seq at this point. The other data we're just getting around to generate now. It got held up by the pandemic. But interesting enough, you can pretty nicely separate your control individuals from your pesticide exposed individuals, and the transcripts that are differentially expressed have an incredibly high mapping rate to degenerative diseases, cancers, things like that. It's kind of a shocking group of differentially expressed transcripts that are that underlie the model prediction with quite good accuracy of who's exposed and who's not. So when COVID hit, DARPA asked us to turn our attention to trying to generate a COVID-19 blood test, with the idea that there's a intergenerational period here between when you're infected, between when you can shed, that's somewhere between 3 and 8 days, according to the modeling studies that have come out since. When, if you've been infected, you'll have a negative virus PCR test. And the idea was that by looking at host response-based assay in blood, you can find a test that would turn positive earlier and in principle use this if you've got individuals exposed and you're tracing. You could more accurately rule out who's actually infected and who's not, and not need to quarantine everyone who's been exposed. And so, the idea is to basically put this through the same type of analysis pipeline I described with some tweaks for COVID. The problem, especially early, this started back in February, March, was obtaining samples and obtaining data. And so we ended up getting involved in collaboration with UNMC, who had sort of the first samples in the country, because they collected the patients from the Princess cruise that was first infected, although we ended up getting very little information from their sample collection. And then we've supported these two related studies of COVID infected and longitudinal new marine recruits called the CHARM study I'll tell you about. We also ran a high-risk healthcare worker at Mount Sinai, but unfortunately or fortunately, the pandemic just passed in New York, the first peak as we launched the study. So we had very, very few subjects turn positive during the longitudinal study. So the CHARM study is the COVID health action response for marine study. It's led by Andrew Letizia, who's a physician, infectious disease researcher. And it really was planned in April 2020. The first subjects were enrolled in mid-May 2020. The initial longitudinal study was completed by November 15th, and it comprised more than 3,000 subjects, 20,000 study visits, 70,000 biosamples. We filled up nearly every freezer at Mount Sinai. This was unlike MotorPack. Everything did not come in perfectly labeled. We would get boxes of tubes that were floating around. It was quite something to handle, especially in the beginning. Eventually, it became more orderly, but it was adapted quite on the fly. And we actually had to field a team of 8 to 10 research and nursing people to support the program in South Carolina. So it's based on the Marine Corps Recruit Depot in Parris Island, South Carolina, which is where all the Marines, all females in the US train there, and all of the recruits east of the Mississippi. We have 300 to 400 recruits arrive weekly for 13 weeks of very, very arduous basic training. And the Navy has a number of large COVID outbreaks. The Theodore Roosevelt received a tremendous amount of national press in March and April. But what got less attention was that Parris Island was shut down by a coronavirus outbreak in March and April, which for the military, they absolutely don't want their pipeline of training Marines to stop. And so this was an enormous wake-up call, and it led to them implementing a extensive and elaborate mitigation plan, and they're interested in launching this longitudinal study to give them some more information. So what the Marines put in place following that March and April outbreak at Parris Island is that all new recruits spent 2 weeks on home quarantine. Then they would arrive to a dedicated location. So through July, they took over the Citadel, which is a military college. It was closed, and they used it purely for the purpose of this 2-week strictly supervised quarantine with very strict control measures put into place. And August through October, it was done at the Sheraton in Atlanta because they reopened the Citadel. After the 2-week period, there would be a Navy mandated PCR test. And in order to go on to Parris Island for basic training, they would have to test negative. And the idea is that they cleared Parris Island of the outbreak from March to April. And the idea of this was they would try to maintain Parris Island as a bubble free of COVID to proceed with the training. As we'll see, it didn't quite work out as planned, although the training managed to proceed. So the CHARM study was built on top of this, this Navy mandated structure, and enrollees in the study would get, during the 2-week quarantine, week 0, week 1, and week 2, enrollees, we'd handle the PCR testing. So it'd be clinical EUA approved SARS-CoV-2 Thermo Fisher PCR testing, blood sampling, questionnaire, temperature check. And then after they moved to Parris Island, 2 weeks after there, so week 4, week 6, and week 8, the same procedure would pertain. In addition, subjects who tested positive would go into be offered enrollment in a separate COVID positive sub-study that would have more frequent sampling. So we did have a second group of samples, and as part of the sub-study, we actually also enrolled 148 Marines who had been in the March and April outbreak. And so this was in May when they first enrolled in the CHARM study, or this COVID positive arm of the CHARM study. So we did a long-term follow-up in them. But mostly we're going to focus on this longitudinal group arriving for 2-week quarantine and then for basic training. So what were they like at baseline? Just under 1% was seropositive, 28 individuals were infected when they arrived, 28 were actively infected, PCR positive, 9% was seropositive. Seropositivity, as you might expect, related to areas of the country, with New York and Louisiana having the highest rates of seropositivity. Most of these subjects were males. Especially from the Eastern, I mean, 7 or 8% were females in the study population. If you look at the cumulative rate of SARS-CoV-2 positivity reported, you can divide your states into sort of 3 groups: the early spring group, with New York taking the lead, the late spring group, and the summer group. And if you look at the cumulative seropositivity of the recruits, you can see that here's the cumulative SARS-CoV-2 positivity from these regions. And in the solid line is the seropositivity with a 3-week running average. And it tracks pretty well with the rate of infections, cumulative rate of infections in those areas for these 3 arbitrarily constructed regions of the country. In addition, Hispanics had the highest rate of seropositivity, and they maintained that through the study. So by the end of the study, they're starting to approach 30% of Hispanic recruits were seropositive on arrival, and the differences were highly significant between Hispanics and non-Hispanic recruits. So now we're going to turn to the first 2 weeks of supervised quarantine at the Citadel and look at what happened there. So the Marines mandated extensive non-pharmacological interactions. They had directional traffic movement, mask wearing at all times, hygiene, social distancing. Everything was controlled. They kept them outdoors as much as they could. And so we studied with the weekly PCR test what happened during this supervised quarantine. And as we mentioned, 16 of this group were positive on arrival, 24 were positive at 1 week, and 11 positive at day 14 additional. And then out of the non-recruits, there were 1848 Marines enrolled in the study, and 1554 were either not eligible or declined. They get tested at day 14, 26 of them were positive. From the charm subjects, we sequenced 36 viral isolates from 32 individuals. If we look at the clinical characteristics, almost none of these subjects were symptomatic at the time of diagnosis. And absolutely nobody either, all of the Marines had daily temperature checks, and it detected 0 cases. So out of the 51 cases, out of the 77 cases that were identified, they were all found by testing done at specific times, scheduled testing, and none of them were found by symptom screening or by temperature checks, which was at the time, that was common practice in colleges and workplaces, and at least for this population of young, healthy, mostly males, it's got no yield. So that's summarized here. If we look at the phylogenetics of the virus isolates, there are a few scattered individuals, but we had shared sequences that represented monophylogenetic clusters. There were 6 of them identified. So these represent single case introductions that then are spreading under this quarantine condition. And if we look at the epidemiology, most of these clusters were associated with either a roommate or being in the same platoon. And so those were the major risk factors. And it turned out two of the clusters happened to occur in the same class, and a lot of them were actually on the same hallway. So it shows an interesting illustration of what contributed and what did not. So the blue individuals here represent the cluster 5 strain. The black individuals are subjects we don't have sequence on, but they were infected, and those are their room locations. To the left here is platoon F, to the right here is platoon E. And what we see is, first of all, there's tremendous co-infection within rooms, and within platoons, the same strain is preserved within the same platoon, with the exception of these 2 individuals from platoon F and platoon E who share the same room. So this is not jumping other than sharing a room from platoon to platoon, so we can eliminate shared bathroom, other activities. It's group interaction within the platoon, and sharing roommates are the major risk factors for transmission here. Okay, so that's what happened in Citadel. What happened in Atlanta? Atlanta, they just left them in the rooms, and there was a very small transmission within roommates. But other than that, transmission rates during the quarantine were much lower in Atlanta. Same number of cases on arrival. Then after their 2 weeks, all of the recruits have to test PCR negative, and then they travel to Parris Island. This is a picture of how they were handling things at Parris Island. So they're introducing mask wearing, other measures. And if we look at, for example, other infections to historical averages, here's the March and April outbreak at Parris Island. And they started introducing a variety of measures here, social distancing, hygiene, mask wearing, etc., isolation of the companies from each other. to try to control the outbreak, which they did. But what we see here is just the respiratory infection rate, and in blue is the historical average. And here we see what happened in 2020. Probably this very high level early is including some COVID cases. And most of these cases in the March and April outbreak were not PCR diagnosed. They didn't have much testing available, so they actually only had a couple dozen cases were confirmed by PCR at that point in time. But the rate of respiratory infection plummets and in general stays well below the historical average. So their intervention is having an effect. If you look at pneumonia, respiratory inflammation, you see the same pattern. It plummets after the outbreak and stays very, very low compared to historical averages. And now this is not the study data, this is the medical surveillance data, but if we look at the COVID case rate, it's enormous after these letters up here mark the various interventions that they've introduced to suppress COVID infection. And despite that, they had a very high number of COVID cases. Here's your respiratory infections. Here we're plotting it year by year. And here's 2020. And these are the COVID cases, which are on a much higher scale that's occurring during that period. So despite interventions that are suppressing other infections, they're getting transmission of COVID-19. So what are the numbers like? We followed about 2,400 Marines who were followed into Parris Island, had not previously had a positive infection, and stayed in the study until their first Parris Island PCR test at 4 weeks of the study, and a little below half of them tested PCR positive during the 6 weeks observation on Parris Island. And we've sequenced now more than this, but we sequenced over 600 of the infected individuals and about 700 isolates. This summarizes the strains and substrains that were identified. So there were 5 clusters identified, and you can see they sort of come in waves, and then they break into subclusters coming over time. So it's essentially every case, with just a couple of exceptions, are members of these 5 introduction events. One of them was one of the clusters we had identified in Citadel, and it carried through, presumably in one of the non-study members who was infected, tested negative at day 14, but was infected at that point and brought it in and spread it. One of them came from South Carolina. We don't know the origin of the other ones. So if we plot all of these on a median joining network graph, we can look at how these various strains are evolving and moving from company to company. So this is a little complicated figure. So the size of the circles are related to the number of cases, with the largest circles being about 60 cases or so. The numbers are the companies, and the companies are numbered roughly in order of when they went through Parris Island from June into October. And so you can see in the most of the cases are P1 and P2. So these are single introduction events that all these are related. but we can see them evolving. So you start in the P1, starts in June, it's in company 1 and 2. Then it mutates to a substrain, 2 substrains, B and C, that both are in company 3. So it's kind of a bottleneck, and everyone here has either strain B or strain C. Presumably it was brought across as a variant and then it mutated within company 3. And then it, when it spread to companies 10 and 13, it's now collected another mutation. So it's strain D, and in time this is moving as well, June, July, and August. And in P2, we see similar, the initial strain shows up in July, goes into, it's in 3 companies, goes into 2 other companies in August as strain B, P2B, P2C, and then P2D out here in September. 4, 5, 3, 4, and 5 are smaller and more restricted in terms of companies, and there aren't clear substrains defined. So Daniel Park and with Jessica Metcalf and Brian Grenfield at Princeton modeled the transmission dynamics in each of these companies. And so this is an SEIR differential equation model, and the parameterization is selected randomly from a distribution of the parameters. So that's what gives the variation and estimates of the reproductive rate from this. And so the data fits for all of the companies pretty well. And the reproductive rate looks within error fairly consistent across all of these. I mean, some of the companies just have no cases, so they fit with very low number, but in general, it seems as if the reproductive rate is reasonably constant. The differences in the numbers are largely attributed to super spreader events, but there is, in general, a bit of an underestimation here of these curves. And the reproductive number, this is a stochastic model, is a bit underestimated from what's observed by the model. And this can be attributed, so if you look at the initial number of exposed individuals, the data fits better that more than one individual is exposed initially. So what this is consistent with is that these companies couldn't leave Parris Island and they were isolated themselves. So what the data is reflecting and the modeling is reflecting is that the introduction occurred from individuals who were either staff or drill instructors that were outside of the companies, interacted with more than one company, and also were allowed to go off of the base. They brought it in, and so the initial, in general, the T0 case of the members who were in the study, the company members themselves, involved more than a single individual being exposed at that point. So the T0, the actual individual bringing in the case came from outside the company. So it's consistent with the data, and the modeling are consistent with what we know of the structure of this outbreak and the epidemiology, and also why it's so cleanly moved in block and with specific mutations from company to company. So what's happening here is, when it's jumping from company 1 and 2 to company 3 and the mutation is changing, presumably there's a drill instructor or some other personnel interacting with this company, picking it up, it's mutating and then bringing it on so it's completely changed when it's introduced to the next company. And we see the initial infection of these companies is typically more than one individual from the modeling. So Daniel also modeled what sort of testing regimen would be effective at suppressing this. So if getting above the, this is the proportion of transmission prevented. If you get above the dotted line, you'll keep the infection from propagating. And what he's comparing here is the frequency of testing on the bottom, the mean generational interval, which in the literature varies between 5 and 8 days. And in our data, it looks to be more like 8 days, but most of the studies have it at 5 to 6. That's the time between when you were infected and then when you transmit it to someone else. That's the generation interval. And then the number of days it takes to get back the test result. And so what we see from this analysis is that more frequent testing is critical, and having a rapid turnaround is critical. And this is the accuracy of the test at the top, sensitivity between 50% and 95%, and sacrificing sensitivity has a less drastic effect. So that if you wanted to suppress these infections, doing something like a antigen-based lateral flow test that would give you an very rapid readback and could be administered on a daily basis, and then you isolate the people who are infected, would enable suppression of the infection. Whereas other testing approaches, you could decrease the number of infections, but you're not going to eliminate the propagation. So if we look at the P1 cluster, when we go from P1A to B, C, and D, this is A, this is B, C, and D. B, C, and D all share a single mutation of serine 3883 to alanine 3883, which ends up being a serine that's highly conserved across the coronavirus's larger family. It's located in the NSP7, which is a subunit of the polymerase. And it also appears to be associated with a reduction in the number of symptoms. The recruits who were infected with the S variant seem to be more symptomatic than recruits that are infected with the alanine variant. So this may be the Marines' attempt to generate their own live vaccine. They have what appears to be an attenuated virus that's spreading when they get to version B, C, and D of the P1 cluster. So long-term follow-up has just started, and this is now 6 to 9 months after study enrollment, and we've analyzed the first out of the recruits who had no infection and were seronegative initially, and through the initial part of the study. We've looked at 135 individuals. Now the team has to travel around the country to the bases where they are stationed. They've left Parris Island to run the study. So we've got the data back on the first 135 of uninfected, and 75% of them are now seropositive. So this suggests that roughly 85 to 90% of the new Marines have been infected by SARS-CoV-2. So it's, interestingly, in the downstream locations, they're not seeing any infections. So they have clearly achieved herd immunity levels from natural infection, possibly the only group in the country that's managed to do that. Okay, so we also did an analysis of reinfection rate. So we can take the baseline serology, and we're using a very stringent test of being seropositive. They need to be 1 to 150 titer or above on both a full spike protein ELISA and an RBD ELISA titering. So we had 225 were baseline seropositive after we eliminate anybody who was infected, et cetera. And 2851 were baseline seronegative. And we can follow these to see how many were infected in Parris Island. And we have an infection rate of 10.1% in the seropositive group and of 48% in the seronegative group. So the relative risk is 0.18 in the seropositive, the previously infected Marines. So we know that these seropositive individuals have gone through home quarantine, 2 weeks of Marine quarantine, 3 negative tests, and then another 2 weeks before the next test. So this is not a persistent infection. The seropositivity is 4 weeks, ascertained 4 weeks, at least 4 weeks before their positive PCR, and they've had at least 3 negative PCR diagnostic tests before they turn positive. And the incidence is of course extremely high. It's 6 cases per year in the seronegative, 1 case per year. Excuse me, rate of 6 per individual per year, and a rate of 1 per individual per year in the seropositive, and a ratio of 0.18 is the risk. Confidence intervals 0.11 to 0.28. The viral CT levels were a little bit lower, but it's not very dramatic in the seropositive group. Symptoms were less, and duration of symptoms, duration of how long they were PCR positive was less, but it was not statistically significant. And this is the KM plot showing the attack rate over time in the seronegative and the seropositive group. So in the seropositive group, almost all the infections were in the individuals with lower titers, 1 to 150 or 1 to 450. That's shown here. And the neutralizing antibody run by Alex Bukarev's lab at UTMB, they were the seropositive individuals who were PCR positive were much more likely to not have neutralizing antibodies. This is a very significant decrease compared to, we tested 54 sequential subjects who did not become infected during the study and started seropositive, and they have a much, much higher rate of having neutralizing antibodies. We also found that the reinfected individuals can shed viable virus. This is from culturing the virus, and out of the 19 reinfections, we had samples on 16, and we could recover live virus from 4 of them. 12 of them we couldn't. So, this is a pretty high reinfection rate, and this is also with the same strains. This is not the British variant or the South African or Brazilian variants. This is a reinfection rate with the same strains that were prevalent earlier in the year in the United States. Okay, so finally I'm going to turn to studies of the host response signatures. This is the pipeline we followed for developing these signatures. So we were emphasizing blood transcriptome to try to get a test that could be more rapidly deployed. We would integrate other data sources such as ATAC-seq or pathway information into the signature selection, and as well as public data that was curated on non-COVID exposures that might be confounds. All of the data use was split in three for training, validation, training, development, and then validation studies. And the signatures were selected by a multi-objective fitness, where we would try to optimize the identification of the COVID exposure, the similarity of the signature via projection. We'd represent the signature as a vector, and we'd look at, for example, attack-seq from COVID data, which genes were identified, and try to optimize the projection of the attack-seq onto the COVID. Or and then we do not want to be identifying non-COVID viral infection confounds, et cetera. And then after genetic algorithm to identify the best signature for this fitness function, we then would work on optimizing it on a clinical platform. And it's taken a while, but it's actually very close to FDA submission at this point. And also characterizing the biology underlying the signatures. So I'll just show you a little bit of that. So this is the specificity data compendium that Daniel Chawla and Stephen Kleinstein's group at Yale assembled. Very large number of samples from many studies that we could use to assess signature specificity. This is the signature fitness function to involve optimizing COVID sensitivity and penalizing cross-reactivity with other viral exposures, other infections, other comorbidities, and to also optimize the projection with other types of data sets. Then went through a genetic algorithm to optimize the fitness value. And eventually we came out with an 11 gene signature from this that we show here, for example, looking at specificity against all of these other studies. And it's showing the signature shows not much cross-reactivity with other confounds. And then if we take the signature and we look at pseudo bulk, it's still showing discrimination in RNA, single cell RNA-seq pseudo bulk data. So that can be used to then look at what's driving the various components of the signature. So shown here, for example, is one of the genes PIF1. And we can show that if there's a single gene knockout in plasmablasts, we can degrade the performance of the classifier most dramatically if we've knocked out PIF1, and also if we knock out PIF1 in the plasmablast, we have a similar effect. So this identifies PIF1 as marking a subclass of plasmablasts that the signature is relying on. And so using this type of approach, we can get a handle on what's driving the underlying biology of the signature. Okay, Frank Zhang and Olga's group has also been looking at alternate splicing for signature development. So the study, we can break our RNA-seq samples into, here's what could be a complete collection. Often we're missing some samples in individual subjects, but the first is when the subject first becomes PCR positive. A pre-sample is in the 2 weeks before. We're kind of interested in these samples because some of these subjects have been infected, but their PCR test has not turned positive. So these are subjects who have a negative PCR test. It's an RNA-seq taken in the 2 weeks before they first turn PCR positive. Earlier samples are control. So these are more than 2 weeks before they develop their first PCR test. And then we have the ones that are following, which we call mid. and then when they turn negative, we call post. So Frank has used a model to identify 4 types of alternate splicing events that are summarized here. The most common is skipped exons. These are at an FDR of 0.05. So most, the largest effect is in first and then in mid, as might be expected. And if you look at this over time, the upregulated alternate splicing versus downregulated alternate splicing, you see that the biggest change is at the first time in which the subjects are PCR positive. This is a summary of all of the events. And so we get a nice time course. It persists through to when they're PCR positive and drops back towards baseline. Post-infection in both the upregulated and downregulated alternate splicing events. If you take the best represented Go terms, neutrophil degranulation is included, and also regulation of alternate splicing. You look at the percent of genes showing alternate splicing. Again, you see a very similar representation of the percent of each of these 4 Go terms that are showing alternate splicing through the course of infection. So in order to see if you can reliably detect infection, we trained on the CHARM data to build a model, and then to test, we used a separate cohort that was collected at Duke, which is an RNA-seq study that they've run outpatient and inpatient there, mostly outpatient. And if we look at the cross-cohort accuracy, we get better results at an FDR of 0.001. So we apply that FDR and we can see we can get a cross-cohort accuracy of about 0.84 AUC, which is pretty good. And we show 2 examples, JLNS and LST1. Both of these are immune-related genes, and you can see that the exon use is going up in CHARM, it's going up in Duke, and here the LST1 is going down in CHARM, it's going down at Duke. So these changes are nicely replicatable and can support predictive modeling across cohorts. Then if we turn to the more difficult problem of can we see infection before the PCR has turned positive, this is going to be a mixture of individuals who are infected but have not yet turned PCR positive, or individuals who are not yet infected. So, the earliest, since this is over a 2-week period, these samples, and you will normally turn positive within a week of infection. So a number of these are just not yet infected. Some of these are going to be infected. So in order to deal with that probability problem, what Frank did was just compare the model that was trained and its prediction of the probability of infection on the control samples for these same subjects versus the pre-samples. And what we see is there's a significant difference between the control and the pre. It's p 10 to the minus 6 in terms of the probability of infection. And there are a few of these samples, these 3 here, that have quite high probability of being infected from the model. If you look at 2 alternate splice groups, LST1, 2 alternately spliced exons, LST and LST1 alternate splicing event, you can see it's starting to go down in the pre on average, and of course drops more in the first, then up again in the mid, and similarly SRSF3, it's starting to increase on average in the pre-samples. The dashed blue line is the control levels. So this suggests that the alternate splicing can pick up evidence for infection before the PCR has turned positive. The other aspect that Frank has looked at is sex-specific changes. I'm just going to show one example here. Genes, altered splice events that are male or female predominant. This is a PTP, which is actually CD45. So it's a T cell marker. It doesn't change in males, but it goes up in females. You can see here in the sashimi plots, you can see this exon is becoming more heavily used in the first sample. Excuse me, it's in the males, it's not really very dramatically changing. It's about the same. In the females, it's very low in the control. And then in the first sample and in the mid sample, it's significantly increased. So this is a hubby gene in the Transgaia human net analysis, which tells you the linkage of the gene to other transcripts via a Bayesian, trained Bayesian network that they've generated. So it's consistent that with the formulation, this is still under study, but that what we're picking up here is a subtype of a T cell identified by differential CD45 splicing that is being altered. Its level is being altered in females only and not in males with infection. So I'm not going through some of the other things. I'm just going to mention them very briefly here at the end. We studied a hamster model of severe COVID and single-cell RNA-seq in response to different Moderna vaccines with Alex Bukharov's group and with Moderna. We looked at the antibody responses to the March outbreak that we started following in May. And we've also done a study with Steve Kleinstein's group where we find that if we do B cell receptors, single cell sequencing, the repertoires in the mild subjects compared with severe subjects are quite different and consistent with a less effective response for generating a sustained immune response in severe COVID. And I mentioned many of the people involved in these studies. They've taken, work's been done by a large number of very, very talented and committed folks working under incredibly hard conditions to get these studies completed. Thank you very much.
Thank you, Stuart. That was quite a journey across 1 year. I'm impressed with all the, you said it at the end, I'm really impressed at how much work got done and how much you were able to produce, under the conditions. I don't know how long, you probably weren't shut down very long or at all, but it's really impressive. So I'll open up the questions and while people, if they want to just chime in, but one of the things that struck me that I'm really interested in was this reinfection data that you had. It suggests that 10% of the people, of these young individuals who were infected originally were able to be reinfected. Do we know that they really had, we assume that they had cleared their infection and then got reinfected. Was there any assessment of the variants? Did the reinfection happen with a different variant than the original infection?
Well, we have the reinfection. The reinfections were caused by the same variants that we have five strains going through Parris Island.
Right.
The reinfections were those 5 strains. So it's coming from its transmission on Parris Island. It's and those strains are, they're not going to be identical. I mean, they are unique at this point in time. They're not identical to whatever infected them before they came. We're identifying them by being seropositive on arrival. So we don't have sequence from their first infection.
Gotcha.
We have one individual who got infected twice during the study, and we have both his sequences, and they're different. But that person isn't counted in the study because this study was just on the people who were seropositive on arrival and then later got infected. But we don't think there are dramatic strain differences here. We expect the reinfection with different strains is going to be much higher. I mean, the evidence from Brazil is there's almost no protection with the predominant variant going through now with what previously, saturated the Amazonia area. So the reinfection risk is huge in this disease.
Yeah, and it goes back to both the herd immunity, but the vaccinations. Are the vaccinations going to be as effective? It seems like the vaccines tend to be very effective at knocking down most of the other variants, right? The UK variant seems to be covered really nicely by the current vaccines, but then, but would this data suggest that either a natural infection in young people don't mount a good response, allows them to be reinfected, and will we see the same 10% reinfection rate in individuals who have been vaccinated? Is there any way to extrapolate the findings of what's out in the literature?
Vaccination has developed so much more effective immunity. The virus has evolved, mechanisms to suppress the development of immunity, and so it's with vaccination you're getting all of the immune epitope exposure with none of the virus's evolutionary adaptations to shut down your immune system. So I do think, I mean, it's not a 10% reinfection rate. It's really close to a 20% risk. So comparative risk. So it really depends on the number of infections you're having. But I think the vaccine certainly will be less, and even with the variants should be less.
Okay, thanks. Anyone else? Josh?
Yeah, I had a question. Stuart, thanks for a great talk. I was curious about the example you mentioned briefly at the beginning where you had some single cell data from MRSA and MSSA infection. Is it possible to tell for a single cell whether it came from MRSA or MSSA, or did you have to kind of average the cells per donor?
I mean, we've done a lot of different perturbations of that comparison, but it's not a huge number of samples. In the data I showed you, it's a pseudo-bulk analysis of the RNA-seq and the attack-seq. We've also done cell type identification and proportion differences and using them to classify. You can do that as well. But we were hoping to get some more samples and expand the numbers. That hasn't proven possible. So the data I showed you was based on a pseudo-bulk analysis, although pseudo-bulk from single cell data is kind of better data than bulk data by itself.
Sure. Yeah. I just wondered, especially since you have such a small number of donors, whether you might be able to get more statistical power if you look at each cell as its own classification problem.
How are you structured? How exactly?
So let's say you have 10 MRSA and 10 MSSA donors. Instead of using 20 data points, treat each cell as its own data point. And let's say you have 10,000 cells from each donor. Now you have 200,000 data points instead of 20.
Okay, we haven't tried that.
Yeah, I'd be really curious to know if that gives you anything different.
Or at least do it within cell subtypes.
Yeah, because within, with single cell technologies are so new that I think exactly how you can use them in these kind of clinical settings is really an open question.
Okay. Anybody else have a question? Because I have another one. This is also going back to early in your talk. You said the seropositive rate tracks really close to the infection rate within each one of these states that you looked at, right? So if we can we get a good idea of how close we are to herd immunity if we took a random sample of people across the state of Michigan and see how close we are? Because there's been there's been a lot of editorials saying, oh, we're so close to herd immunity already, but then some other studies would suggest that we're only at 40%.
Chuck, the herd immunity is the level. Herd immunity depends on your virus reproductive rate. The virus reproductive rate depends on the environment. So what herd immunity is there's not a fixed number for herd immunity. It depends on the environment. Probably in rural areas with less contact, it will be lower. In cities, it's going to be higher. It depends on how much interaction you have, how much exposure you have, because you can dramatically change the reproductive rate by putting measures in place. For example, it changes it, and herd immunity is just, for example, in these Marines, the reproductive, we're seeing a reproductive rate of approximately 6, which is enormous compared to what's been reported in any study except for the outbreaks on ships. So they're acting like a ship outbreak, and that requires 90% to get herd immunity because the reproductive rate is so high. If you have a lower rate, so for example, these variants, aside from the fact that they can cross-react and reinfect and all of this, if they have a higher reproductive rate, because they're more infectious, they also require, just from that alone, a higher rate. So you can plot it out. They're directly correlated. So there's absolutely no answer there.
Well, so I understand that, but if let's take an example. You take Ann Arbor, and if we took a random sample of 500 people in Ann Arbor, we're sort of a herd, right? And it's close. It's a community represented. If you look at the seropositive rate in this community, because the seropositivity tracked with the rate of infection at any given time, or does that no longer apply since a lot of people have been infected already, and that's changing this curve? Because very early on, the seropositive rate tracks with the rate of infection in the communities that you looked at, right? Sure. So maybe I'm misunderstanding it, but can we estimate some things by random sampling?
The seropositivity is basically it's the cumulative infection rate. The seropositivity is just an integration of the infection rate over time. Anyway, I don't know the answer to your question.
Okay.
I don't know what level is going to give us herd immunity, but probably when we get to something like 70%, we're going to be in pretty good shape, I would guess.
Yeah. It also doesn't. If variants can reinfect, the reinfection rate is going to be.
You'll have a mixture. You'll have a lot of people vaccinated, and then you'll have some people who just had natural infection. But I think we're going to get there, and it's not going to be too long.
Okay. I like your positivity. Anybody else have a question? If there's none, thanks a lot. Josh had to step out, but for everybody here, thanks a lot, Stuart. This was it was very cool. I learned a lot today, and good luck with getting all these papers published. It looks like you're going to have 20 papers out of one user worth of work, which is pretty darn good.
All right, thanks, Chuck. Great to see you. Thanks for
everyone. Bye.
Goodbye, everyone.